Hello, everyone. I'm very excited for this opportunity to talk about some of our work evaluating blood-to-brain glucose transport kinetics and GLUT-1 regulation in the human iPSC-derived in vitro blood-brain barrier model. So the blood-brain barrier is fundamentally comprised of brain endothelial cells, or BECs, which line the blood vessels in the brain. BECs have highly specialized properties, such as the expression of tight junction proteins, which confer barrier functions to prevent the unregulated diffusion of bloodborne solutes into the brain. But in addition to barrier functions, BECs also serve as an active interface, mediating the strictly regulated transport of nutrients, proteins, and other molecules in order to maintain brain homeostasis. So blood-brain barrier dysfunction is widely implicated in neurodegenerative diseases, such as Alzheimer's disease, or AD. And one feature of AD is brain hypometabolism of glucose. And because glucose is the primary fuel for brain cell metabolism, this leads to a severe brain energy deficit that plays a major role in dementia and the overall cognitive pathology of AD. So one hypothesis is that the GLUT-1, the predominant glucose transporter at the blood-brain barrier, may be dysfunctional in AD, leading to decreased glucose uptake into the brains of AD patients. So the underlying molecular mechanisms that modulate glucose transport in both physiological and pathological contexts really is best studied in a humanized in vitro model. But unfortunately, most in vitro models that employ either primary or immortalized human BECs suffer from weak barrier properties, which make it difficult to examine transcellular transport pathways without confounding effects of paracellular leakage. So to study this, ideally we need a model that exhibits both strong barrier properties and a glucose transporter expression profile representative of the human blood-brain barrier. So we can actually take human iPSCs and differentiate them into a brain endothelial cell-like phenotype that displays many blood-brain barrier-specific properties, creating a model that is well-suited for studying blood-to-brain glucose transport kinetics and mechanisms of GLUT-1 regulation in human cells. And so we call these cells iBECs. And our first aim was to characterize the kinetics of luminal to abluminal glucose transport in iBECs, which functions to model blood-to-brain glucose uptake. So this entails determining the saturability of the transport system upon saturation with excess glucose, the extent of inhibition by pharmacological inhibitors, and relating glucose transport rates to GLUT-1 transporter expression levels. So to do all of this, we wanted to focus on two stages of iBEC maturation into the blood-brain barrier phenotype in order to examine glucose transport related changes that occur over time as the cells are cultured in vitro. And finally, we aim to identify signaling pathways involved in the regulation of GLUT-1 and glucose transport to investigate potential targets for therapeutic intervention that might improve outcomes in AD patients. And so to create iBECs, we start by seeding iPSCs at an optimized seeding density. And then we initiate differentiation using a defined medium that supports both neural and endothelial progenitors in an environment reminiscent of the developing brain. We then further drive the endothelial cell phenotype using human endothelial serum-free medium, supplemented with basic fibroblast growth factor and retinoic acid. Then by subculturing onto collagen fibronectin-coated surfaces, we select for the endothelial-like cells, and then we do a final medium change the next day to remove the non-inherent cells and the BFGF and RA signals, which leaves us with a relatively pure population of iBECs. And since this is the first day that transendothelial electrical resistance, or TEER, is measured to evaluate barrier function, we term this days in vitro 1, or DIV1. And importantly, the medium is not changed hereafter, and the brain or the barrier phenotype develops without any further medium changes. And so this is what the Transwell format looks like. And this allows us to model the blood-brain barrier by separating the luminal compartment, which models the blood, from the abluminal compartment, which models the brain, the interface, of course, being monolayers of iBECs. And we have developed an assay to quantify the rate of glucose transport across these monolayers, which essentially consists of adding a radioactive glucose analog into the luminal compartment and calculating the clearance in microliters of radioactivity transported to the abluminal side. And when that clearance is plotted over time, the slope of the resulting line can be used to calculate the endothelial permeability surface area coefficient, or PE, for the transwell, which is the quantitative outcome measure for the rate of glucose transport. And we can also measure the PE for DTPA as a low molecular weight leakage control for both paracellular and transcellular leakage. And so here are the results of 3 independent differentiations of iBECs termed experiments 1, 2, and 3, and color-coded accordingly, in which we examined the saturability of the transport system on day 2 and day 9 after subculture onto the transwell format. And so we chose these 2 stages of iBEC maturation corresponding to the 2 time points when tiers are usually at or near their highest points. So we initially see an initial peak around day 2 to day 4, and then a second peak that's generally around day 9 to day 12. So we chose days 2 and 9 for investigation. And we show evidence of a saturable transport system by the significant reductions in PE upon the additions of excess glucose into the luminal chamber, along with the radioactive glucose analog that we use to observe glucose transport. But interestingly, the extent of transport saturation is pretty similar on both days. It's about a 79% reduction on day 2 and about a 74% reduction on day 9. But the baseline rate of luminal to abluminal transport is decreased by 40% on day 9 relative to day 2. And this was interesting to us because we consistently see stronger barrier properties as indicated by higher tier on div 9. So we were sort of expecting a higher rate of glucose transport, but instead we saw the opposite, which is that it was significantly lower. And so in both of these representations, the circles are the unsaturated controls with a normal level of glucose, and the squares are the saturated monolayers with excess glucose to a concentration of 55 millimolar. And importantly, the high concentration of glucose does not show any evidence of leakage, as there's no difference in the DTPA PEs for the saturated and unsaturated groups on either day. So here in clearance curve format for experiment 2, you can see the flattening of the unsaturated slopes that occurs going from div 2 to div 9, which illustrates this reduction of the baseline rate of transport, which is indicated by this comparison here. And so we wanted to explore this evidence that there are phenotypic changes happening during long-term culture that cause a significant reduction in iBEC glucose transport capabilities. And we immediately thought of differential regulation of GLUT-1. And based on our data, we hypothesized that GLUT-1 expression is decreased over time, which leads to the decreased rate of glucose transport on div 9. But surprisingly, we found that GLUT-1 expression actually increases over time, and we show that in 2 ways. So we can immunofluorescently label GLUT-1 and directly visualize and quantify the fluorescence intensity on a microscope. And we can also do a Western blot analysis on protein extractions from div 2 and div 9 cells. And so we clearly see that GLUT-1 expression is much higher on div 9, which seems to contradict with the results of our glucose transport assays. So our results indicate that the high expression of GLUT-1 on div 9 does not translate to a higher rate of luminal to abluminal glucose transport. So we are currently investigating the cause of this disparity by firstly examining changes in GLUT-1 subcellular localization to determine the relative luminal versus abluminal distribution of GLUT-1 on div 2 and div 9. Secondly, we are examining post-translational modifications such as phosphorylation that could modulate GLUT-1 function. Thirdly, we're characterizing abluminal to luminal transport rates, which models brain to blood glucose transport. And lastly, we are examining glucose metabolism differences, which could provide insight on the molecular changes that contribute to brain endothelial cell maturation and preservation of brain energy homeostasis. And if we can elucidate the molecular regulators of GLUT-1 expression, function, and localization, we can inform future studies into therapies for AD that may improve glucose uptake via GLUT-1 into the brains of AD patients. But importantly, we have shown that glucose transport across iPSCs is primarily mediated by GLUT-1 because it is significantly inhibited in a dose-dependent manner by the highly selective GLUT-1 inhibitor BAY876. And we have also identified DAPT as a novel inhibitor of glucose transport in IBECs. So DAPT is a gamma secretase inhibitor that inhibits the Notch signaling pathway, which is a prominent signaling pathway regulating endothelial cell energy production, maturation, and quiescence. So this preliminary data implicates Notch signaling in the regulation of glucose transport and GLUT-1 in brain endothelial cells. So in summary, we have demonstrated that luminal to abluminal glucose transport in IBECs is a valuable model of blood-to-brain glucose transport as it is saturable and inhibitable by a GLUT-1 specific inhibitor. We have also shown that while the rate of glucose transport decreases over time, GLUT-1 transporter expression increases and TIR increases as well. So over time, signaling pathways controlling either GLUT-1 function, localization, or both may be altered in a way that significantly decreases the rate of luminal to abluminal glucose transport. And finally, we have identified Notch signaling as a potential pathway involved in GLUT-1 regulation. So thank you all for listening, and thank you, Puerto Rico Neuroscience, for the opportunity to share our work.